Hello and welcome everyone to today's Revive webinar by the Global Antibiotic Research and Development Partnership, GARDP. My name is Victor Kwasi and I'll be hosting today's Journal Club webinar in which we'll have presentations of key findings from recent publications in antimicrobial R&D. Revive is GARDP's education and outreach program. It aims to connect and support the antimicrobial R&D community by facilitating learning, sharing knowledge, and connecting people. These webinars are part of our educational activities. All of our webinars are recorded and are freely available to view after the live broadcast on our website, revive.gardp.org/webinars. You can also read our series of articles known as Antimicrobial Viewpoints, in which experts discuss various topics in the field. And we also have a resources section that includes the Antimicrobial Encyclopedia. This has definitions on various important terms that you'll hear about in today's webinar. Some of these also include videos in which experts give further explanations on the terms. As always, today's presentations will be followed by a Q&A session. You can submit your questions at any time during the webinar by clicking on the questions button as shown on the slide. We will address the questions after the presentations and do our best to respond to as many as possible. Today's speakers are Melis Anahtar, Nicole Scangarella-Oman, and David Paterson. Unfortunately, Nazgul Sakhanova will not be able to present today. Our moderator today is Florian Maurer. Florian Maurer is the head of clinical development at Basilea Pharmaceuticals International AG, Switzerland, and a professor of medical microbiology at the University Medical Center Hamburg-Eppendorf. Holding both German and Swiss specialist certifications in clinical microbiology, his professional focus is the development of novel anti-infectives and diagnostic assays to manage infections caused by highly drug-resistant bacterial and fungal pathogens. His experience includes leadership roles as a global clinical development director, infection and immunity at Roche Diagnostics, as the head of the supranational reference laboratory for mycobacteria at Research Center Boston, Germany, and as the director of the Antimicrobial Stewardship Program at the University Medical Center Hamburg-Eppendorf. He previously also held leadership positions within WHO and ESCMID advisory and steering groups dedicated to combating drug-resistant infections. Welcome, Florian. I now hand over to you to introduce our first speaker today.
Thank you, Victor, for the kind introduction, and also from my side, a warm welcome to everybody joining us today on the line. I understand we have quite a lot of people joining us today, so I think that is a testimony to the presentations we will hear and also the great speakers we will have here with us today. So really great to have you all with us here, and I'm really looking forward to these 90 minutes we will have together. We will roughly have 15 to 20 minutes time for each presentation, and then, as Victor mentioned, we will have 5 to 10 minutes for a Q&A after each presentation. So I would just like to reiterate that you please make use of the Q&A function, and then I will try to curate whatever comes in so that we can cover as much ground as possible. So that being said, without further delay, let us begin with the presentations right away. And I think our first speaker today probably is well known to many of you. However, David, of course, still deserves a proper introduction. So our first speaker today will be David Paterson. He's the Director of Advanced ID at the Saw Swee Hock School of Public Health and the Yong Lu Lin School of Medicine at the National University of Singapore. Advanced ID is a clinical trials network that comprises more than 100 hospitals across 20 countries in Asia. And this network is jointly funded by the Wellcome Trust and a number of Singaporean institutions with an aim to conduct clinically important trials in the field of AMR. This comprises trials of antibiotics, diagnostics, but also prevention strategies. David is also an honorary professor at the University of Queensland and has more than 600 peer-reviewed publications, predominantly in AMR. His research focuses on the molecular and clinical epidemiology of infections with antibiotic-resistant organisms, with the intent of translating knowledge into optimal prevention and treatment of these infections. Multi-country clinical trials are the major component of his research portfolio and the predominant focus of Advance ID. So welcome, David, and really great that you find the time to join us today. And please start your presentation now. Thank you.
Thanks so much for the introduction, Florian, and a big thanks to GARDP for hosting this journal club. You'll see here that the background of my slides is the University of Queensland, even though I'm working now in Singapore. And the reason being is that the University of Queensland was the sponsor of this trial called the Game Changer trial. So I do have some disclosures, including that I am a consultant to GARDP and that the Game Changer trial came as a result of a grant from Shionogi. So I'd like to first, in the spirit that this is a journal club, just provide a little plug for a newsletter that Advance ID puts out every 2 weeks. And the reason I wanted to mention it is that we survey the literature related to bacterial and fungal infections. We provide a little summary, particularly when there's randomized trials. It's a completely free newsletter, and if you would like to subscribe to it and have it come directly to your inbox every 2 weeks, please feel free to scan this link. Sorry about the little advertisement, but hopefully you'll find the information potentially of interest. But today I'd like to talk about this randomized trial that we conducted, which was related to the antibiotic Cefiderocol. And just as a little piece of background information, it's important to know a little bit about mechanisms of resistance of gram-negative bacilli. So when we talk about beta-lactam antibiotics, what they really need to do is get down and bind to penicillin-binding proteins. But to get there, they have to encounter the periplasmic space where beta-lactamases live, and also on the inner aspect of the outer membrane, there are some of our nastiest beta-lactamases, particularly NDM. But even before they get to that, they need to get through the outer wall, the outer membrane of the gram-negative bacillus. And that can be a very difficult ask. So what was quite ingenious in terms of the development of Cefiderocol is that the bacteria actually thinks it's taking iron through the outer membrane. But unfortunately for the bacteria, it gets surprised because the antibiotic is what we call a siderophore cephalosporin, and the bacteria is actually bringing in an antibiotic, which it thinks is actually iron, through the outer membrane. So this is an ingenious mechanism of action. And I remember going to some early meetings on Cefiderocol where we looked at its spectrum against gram-negative bacilli, and really it was an amazingly effective antibiotic in vitro against wild-type gram-negative bacilli, whether they're Enterobacterales, Acinetobacter, Pseudomonas, or Stenotrophomonas. The initial clinical trial was also extraordinarily successful. This compared Cefiderocol with imipenem for complicated urinary tract infections. And even though the trial aimed to show non-inferiority, not only did it show non-inferiority, but it also showed superiority. So luckily, when I was cleaning out one of my computer bags, I found some notes when I'd first talked to Shionogi about the potential trial. And here we have an antibiotic that is extremely active against gram-negative bacilli. In its first clinical trial, there was superiority over standard of care. And I was trying to sort of come up with some names for the clinical trial. And you can see one of them there, amongst breakthrough and leap forward, was Game Changer. And so, in fact, we called the trial the Game Changer trial, and that was the name that we used internally, in both internally and externally, for the clinical trial. So the Game Changer trial was an investigator-initiated trial. Victor, maybe if you can advance, because for some reason it's not advancing for me. It was an investigator-initiated trial of Cefiderocol versus standard of care antibiotics for both hospital-acquired and healthcare-associated gram-negative bacterial infections, particularly, well, exclusively bloodstream infections. So just going through some of the methods, and please advance, Victor. First of all, I'd like to sort of go through the methods and then come to the results and some discussion points. Next slide, please. So this was a trial in which we set out to show non-inferiority, but secondarily, if we showed non-inferiority, we looked for superiority. We evaluated Cefiderocol with its standard dosing regimen, although also allowed for two things. One is to go up with the dosing regimen if there's augmented renal clearance, ARC. And secondly, we provided the sites with discs, and they could use a second antibiotic in combination with Cefiderocol until we'd proven Cefiderocol susceptibility. Next slide, please. So a randomized trial, Cefiderocol on one arm, and the other arm it was standard of care antibiotics. And this was completely up to the clinician at the site. They could use up to 3 antibiotics with activity against gram-negative bacteria, and they could change the antibiotics as they saw fit. In both arms, you could have drugs also active against gram-positives, anaerobes, or fungi. Next slide. This was a non-blinded trial. And next slide. I'd like to talk about the inclusion criteria. So basically, patients were enrolled if they had bloodstream infection, so a blood culture bottle with gram-negative bacilli. We did not allow inclusion for more than 48 hours after the time of that blood culture collection. So they had to have no more than 48 hours elapsed. It had to be hospital-acquired or healthcare associated, and we restricted it to adults. Next slide. In terms of exclusion criteria, we didn't want to have patients who were about to die. Clearly, we didn't want to have patients who were allergic to cephalosporins, significant polymicrobial infections, a line-related infection with the line not able to be removed, and you can see some other exclusion criteria there. Because it was an investigator-initiated trial and not a trial for regulatory approval, we could be very pragmatic and tried to minimize exclusion criteria so that we could have as many patients as possible in the trial and provide it to be as generalizable as we could. Next slide. So basically, patients were randomized 1-to-1 to either Cefiderocol or standard of care. We did stratify by their comorbidity index and also by the region in which the patients were hospitalized. Next slide. We had as our primary outcome measure 14-day all-cause mortality. And as I mentioned earlier, our hypothesis was that Cefiderocol was non-inferior to standard of care, but we also built into our statistical analysis plan the opportunity to look for superiority if non-inferiority was met. And we used a non-inferiority margin of 10%. Next slide. So we did have a main analysis population which we pre-specified, and that was basically all patients who were randomized except if they didn't end up growing an aerobic gram-negative bacillus. We had to have at least 1 dose of study drug or an agent active against gram-negatives in the standard of care arm, And we did have occasional situations where patients withdrew consent before the primary outcome measure, and so that they were excluded from the main analysis population. Next slide. We also pre-specified another patient population for which, again, we were going to assess for superiority, and this was the carbapenem-resistant subset. So patients who had carbapenem-resistant gram-negative bacilli. Now, I do want to emphasize we did not know at the time of randomization and inclusion in the trial whether or not a patient had a carbapenem-resistant organism, but this was a predefined subset. Next slide, please. So let's now look at the results. And first of all, next slide, if we look at the consort diagram, we assessed over 9,000 patients for eligibility, and many of these were excluded because the bloodstream infection was not hospital-acquired or healthcare associated, or there was more than 48 hours that had elapsed, or the bloodstream infection was in fact not a gram-negative bacillus. Next slide. So we ended up randomizing 250 patients included in the main analysis population in the Cefiderocol arm and 254 patients in the standard of care arm. So let's now have a look at the baseline characteristics. Next slide, please. And you'll see that the baseline characteristics were well matched in terms of demographic variables and also comorbidities. Also, you can see there that quite a large proportion, over 50%, were immunosuppressed, and between 15 and 20% were neutropenic. So again, very much reflecting the sort of populations that we see in clinical practice. Next slide, please. Also, when we looked at the severity of illness and the sources of infection, you can see there that we had quite a variety of sources of bloodstream infection, about 20% urinary tract infection, 15 to 20% intra-abdominal, and you can see down the list. So again, reflecting what really happens with hospital-acquired or healthcare-associated gram-negative bloodstream infections. Next slide, please. So in terms of microbiology, E. coli was the most frequent organism, but very closely followed by Klebsiella, and then Enterobacter. So Enterobacterales, as you can see there, were responsible for about 70% of patients enrolled, whereas the non-fermenters, Pseudomonas, Acinetobacter, Stenotrophomonas, were much smaller in their numbers. Next slide, please. So let's look at the outcomes, and we'll start with the primary analysis population. And again, remember the primary analysis population is every patient who was enrolled, who did grow an aerobic gram-negative bacillus, who did have at least one dose of the study drug, and were able to be followed out to 14 days. So this included both carbapenem-resistant and also carbapenem-susceptible organisms. Next slide. So let's go directly to a table from our manuscript. And you can see here that the primary endpoint was day 14, all-cause mortality, and 8% of those who received Cefiderocol had all-cause mortality by day 14, and 7% in the standard of care group. We had a non-inferiority margin of 10%, and so non-inferiority was achieved. But you can see there that numerically, with more patients in the Cefiderocol arm dying than the standard of care arm, superiority was not met. So let's go down now to the carbapenem-resistant subset. So these are the patients who we found had a carbapenem-resistant organism. And you can see at day 14, 14% in the Cefiderocol arm had mortality by day 14, compared to 10% in the standard of care arm. I said earlier that we were looking for superiority, but again, clearly superiority was not met with a numerically higher percentage in the Cefiderocol treated patients having mortality. Next slide, please. So we did follow the patients out to 90 days, and in the upper Kaplan-Meier graph, you can see that they are very, very tightly matched. The lower Kaplan-Meier graph is the patients with carbapenem-resistant organisms, and you can see there that right up until close to 90 days, there was some gap, a higher mortality in the Cefiderocol treated patients than in the standard of care patients. Next slide. For clinicians, this is the data which I think is the most interesting. And if you start at the top row, MBL positive CRE, so these are metallo-beta-lactamase producing organisms, and the vast majority of these were NDM producers. The 14-day mortality was 31% in the Cefiderocol treated patients, and none of 11 patients randomized to standard of care died by 14 days. So this for us was the most interesting finding and perhaps the most unexpected finding. You can look down there at other carbapenem-resistant organisms. Some of you may have known about the Credible trial, in which there appeared to be an increased mortality when Cefiderocol was given for Acinetobacter. But in our trial, obviously the numbers are small, but carbapenem-resistant Acinetobacter mortality was pretty similar in both arms. Next slide, please. So our, discussion points. First of all, if we look at the total population, patients with hospital-acquired or healthcare-associated gram-negative bloodstream infections, Cefiderocol was non-inferior to standard of care, was not superior. The populations appeared well matched at baseline. This was quite a large sample size. In the subpopulation with carbapenem-resistant organisms, the reality was the proportion was significantly smaller, 126 patients in total. But I think some of the standout points were that MBL positive strains, particularly NDM producing strains, did appear to have a poor outcome when treated with Cefiderocol, but quite acceptable outcomes were seen with carbapenem-resistant Acinetobacter, other types of CRE, also Stenotrophomonas, and carbapenem-resistant Pseudomonas aeruginosa. Next slide. So I'm very happy now to take your questions. I know that was quite a rapid tour through a clinical trial that actually took 7 years from the time we had first discussions with Shionogi to the time it was published. But please send through your questions and I'm very happy to answer those.
Thanks a lot, David, for this very clear and concise presentation. I think this is really super interesting data. Now, as we wait for questions to come in, and I would like to encourage the audience once again to please make use of the fact that we have David here with us and enter your questions in the Q&A. Now, as we wait for questions coming in, and since this is also an educational event here, I would have a question which could maybe start us off, David, not so much regarding the results of the trial, but more your considerations around the trial design. So traditionally in the ID space, we would conduct non-inferiority trials. Here you went for a hybrid design, which could turn into a superiority trial as well. Perhaps for the audience, and I know this is a huge topic, so maybe you can just give a couple of high-level comments around the thinking around non-inferiority trials versus superiority trials and hybrid approaches in the space.
Yeah, no, this is a really important question, particularly if we're considering a new antibiotic that may potentially have advantages over standard of care. And when we went into this trial, we'd seen the in vitro activity, we had seen the results compared to a carbapenem for UTI. And we thought, well, on the other side, we're going to have a lot of colistin-treated patients or polymyxin B-treated patients, maybe a little bit of ceftazidime, maybe Bactam, but we really thought that there was a decent chance that Cefiderocol may have actually been superior to standard of care antibiotics. Now, of course, that was not borne out, and that was an important lesson. But I guess in general terms, non-inferiority is a lower bar to leap over. One of the big challenges, though, is what is a non-inferiority margin that's acceptable if you're developing a new drug to regulators. If you're doing a trial where the audience is predominantly clinicians, what non-inferiority margins would clinicians find to be acceptable? So that's one of the big challenges with non-inferiority designs, is what is the margin going to be? With superiority trials, we don't have that issue. However, we do have, I think it's a lot higher bar to get over, because if, for example, we're looking at all-cause mortality as our endpoint, we know that patients die, particularly if they're hospitalized patients already, not just because of their infection, but because of their underlying disease. And that, in a way, makes non-inferiority much more likely than superiority. And so it's quite rare. It has happened. And even last week, there was a trial in which inhaled liposomal amikacin for Mycobacterium avium complex did leap over that superiority bar compared to oral therapy only. But it is a challenge, and I think we have to be aware that there's a lot of elements that have to go into design of a superiority trial that we don't have to think about as much with non-inferiority trials. So sorry, Florian, we could talk an hour about non-inferiority and superiority trials, but it's a huge, huge important topic.
Very well. Thanks a lot, David. And I think this gave us enough time to receive some more questions. In the interest of time, I will just pick two, because we have to also be mindful of the fact that you will need to leave for another commitment. So the first one that came in was, as the Game Changer trial was not for registration purposes, what was actually the goal that you wanted to achieve? I think you briefly touched on it, but maybe you can just summarize it real quick.
Yeah, I mean, when we have a new drug, it is interesting that often when the trials are done for regulatory purposes, they're not done against the toughest organisms, and therefore they're not compared to antibiotics that are active against, for example, carbapenem-resistant organisms. So, for example, with Cefiderocol, it was first approved for complicated UTI versus a carbapenem. It also was approved for ventilator-associated pneumonia. I'm talking about FDA approval here, versus a carbapenem. There was a trial done only with carbapenem-resistant organisms, but it was what was described as descriptive. It didn't have a non-inferiority margin. So our real purpose was, well, if clinicians are going to want to use this drug for carbapenem-resistant organisms, what are the outcomes going to be? And that's really the purpose of having it with hospital-acquired and healthcare-associated infections and excluding community-acquired infections, because we really wanted to enrich for patients with carbapenem-resistant organisms. So it was really for a clinical audience.
Very well. And then the last question, I think this is really also an interesting one here by Russell. He asks, what percentage of patients had their Cefiderocol doses increased because of augmented renal clearance? And do you think that underdosing may be a possible explanation for the higher early mortality in patients with NDM?
Yeah, even though we had in our protocol, we could increase it. That was actually very rare. And in fact, most patients just had the conventional dose. So I think it's unlikely that underdosing was the reason for Cefiderocol not being superior. I think to me, the biggest reason is that we have subsequently learnt that NDM does compromise Cefiderocol, and therefore its activity against metallo-beta-lactamase producing strains, when given by itself, is actually not as good as we'd initially hoped. And I think that for me was one of the biggest things I learned from the trial, and it was interesting when that's subsequently been matched up with mechanistic data as well.
Very well. Thanks a lot, David, for covering these questions. Victor, perhaps we can capture the other questions that came in, and then we may perhaps find a way to get some more answers here as we collected a couple more, which I'm afraid, again, in the interest of time and also the time for the other speakers, we won't be able to cover right now. For now, thanks a lot, David. I understand you may need to dial out for another commitment. So if that's the case, once again, on behalf of everyone, thanks a lot for joining us today. And let's move on to the next paper. So our next presentation will be given by Melis Anahtar. Hi, Melis. Good to see you on the line. Very well. Melis is a physician scientist whose research and clinical work focuses on developing novel therapeutic and diagnostic approaches to combine the rising threat of infectious diseases. Melis is an assistant professor in pathology at the Harvard Medical School with joint appointments at the Mass Gen departments of pathology and medicine. Melis also leads a research lab at the Division of Infectious Diseases and serves as an assistant director of the MGH Clinical Microbiology Lab. She previously co-founded Day Zero Diagnostics, a sequencing-based infectious disease diagnostic company, which was recently acquired. I'm very happy now to welcome you, and the floor is yours, Melis.
Thank you very much, Florian, and thank you to GARDP for the opportunity to present today. I'm happy to tell you about a paper that was published last October in Cell on a generative deep learning approach to de novo antibiotic design. And this was a project that I was lucky to co-lead with two wonderful lab partners, Arti Krishnan and Jackie Valeri. And we did the work in Jim Collins's lab, who's at the MIT and Broad Institute. And a year ago, I finished my postdoc in Jim's lab, and now I'm faculty at Massachusetts General Hospital in Boston. So I don't think I have to convince this audience that we need new antibiotics to be able to keep up with this ever-increasing rise of bacterial resistance to existing antibiotics. And of course, I'm sure you all are familiar that the first antibiotics that were discovered were from nature, mostly serendipitous. But in the past few decades, the approaches to be able to discover small molecule antibiotics have really ranged on one end of the spectrum from rational design to the other end of the spectrum to high throughput screening. And in the past few years, there's been this in-between approach that's emerged, which uses deep learning for antibiotic discovery. And I'll spend a little bit of time kind of explaining the concept because it might be a little foreign to some of you, but the idea here is that you can train a machine learning model on a carefully designed chemical screen. So it's a small high throughput screen that's somewhere between 1,000 or maybe 100,000 compounds. So the model is trained on that data, and you can then ask the model to extrapolate from that data to identify potential new antibiotics. And this proof of concept that this approach could actually work was published in Cell in 2020 with the discovery of halicin. And halicin is now being developed to treat H. pylori infections. Sorry, let's see if I can advance it. There we go. So to go a little bit deeper, deep learning, as you may be familiar, is a subset of machine learning, which is a subset of the even greater category of AI. And deep learning uses artificial neural networks that have multiple layers to automatically learn representations of data. And the really ultra distilled idea is that we build this predictive machine learning model that's essentially a function. It takes a chemical structure as an input, and it outputs a number between 0 and 1. If it's 0, that indicates that the chemical structure is not predicted by the model to have bacterial growth inhibition. If it's a 1, the model is saying it's extremely likely to be growth inhibitory. The nice thing about this is we don't have to blindly trust the model. We can just order and validate the model's antibacterial activity by physically testing the compound against a bug in vitro. And the hope is that you end up with that picture of a hidden gem of a new antibacterial compound. And sorry, let's see if the slide advances. So we've taken the approach of training these models on phenotypic screens, as in we're looking to see if a small molecule actually inhibits bacterial growth at 50 micromolar in the context of the whole cell. And the idea here is that we are mechanism agnostic, so hoping to find new mechanisms of action. And we've taken the approach of trying to train organism-specific models. So we have 39,000 compounds that we've tested against gonorrhea, 39,000 that we've tested against Staph aureus, and we have separate models that predict antibacterial activity for each of these organisms. And those are the two models that we used in the paper that I'll describe today. Let's see. And the idea here is that the model is kind of like an oracle, where we can show this oracle a small molecule, ask it, will it inhibit, let's say, gonorrhea growth if we're using the gonorrhea model, and it'll give an output of 0 to 1 that corresponds to the oracle's thumbs up or thumbs down. And the nice thing is that unlike a human oracle, this oracle doesn't get tired. It can evaluate thousands of molecules per second, which can prove to be quite powerful. Next slide. All right, so one way to use these models to discover new antibiotics is to just use it to virtually screen commercially available chemical space. Commercially available chemical space is now extremely large. There are about 70 billion small molecules that can be made on demand. That's about 10 to the 11th molecules. And there have been a few research groups that have really exploited this very large chemical space for both target-based or phenotypic-based drug discovery. But that number is really dwarfed in comparison to the estimated number of compounds that are drug-like, which is thought to be around 10 to the 60th. So we wanted to know whether we could kind of use our antibacterial deep learning models and pair them with generative AI approaches to try to design new antibiotics de novo that don't exist in commercially available chemical space, to try to gain even greater structural novelty. And we care about structural novelty because it is a means of gaining access to new targets and overcoming existing antibiotic resistance mechanisms. And these numbers are kind of hard to fathom, so I thought I would give you some intuition about how much drug-like chemical space really remains unexplored. So here's an analogy, bear with me. Imagine if a small molecule is a single grain of sand, and you were to spread out a layer of 10 to the 11th grains of sand to represent the size of commercially available space. That would cover about the floor of 2 IKEAs. Now, what if you wanted to cover the floor of the surface area of the entire Milky Way galaxy? You need 10 to the 48th grains of sand. And 10 to the 60th grains of sand would really take up the entire surface area of the universe. So how do we even begin to bridge that vast gap between IKEA and the universe? So that's what I'll try to describe, how we tried to do that today. So you can imagine there are countless ways to do it. We decided to try 2 different approaches. One was where we provide a chemical seed in the form of a fragment to a generative AI model to give it something to expand upon. And the second approach is just giving the model the most basic chemical starting points and really let it go free, which we call de novo design. And to try to find promising antibiotics from this, we really used our graph neural network models, those models, that oracle that I showed you before, to score the compounds and tell us whether or not we were moving in the right direction. Is it antibacterial or not? And we paired it with 2 different generative models that I'll describe in detail, but briefly, one of them is a model that's based on making chemical mutations, and the second is a variational autoencoder. And then once we had these new compounds that were designed by our generative AI methods and validated by our models as being possibly antibacterial. We then had to have those compounds evaluated by commercial chemical synthesis providers and custom synthesized. And we could then test the compounds directly on bacteria and see if they had those antibacterial properties that we desired. So I'll first tell you about how we did the fragment-based design. Okay, so for the fragment-based identification, we first used our Neisseria gonorrhea model with the hope of finding new antigonococcals. And the first step for fragment-based design was finding, of course, a novel fragment to use as the seed for generative AI. And of course, we all know that fragments like beta-lactam rings and quinolones really underlie the biological activity of a lot of our highly effective structural classes of antibiotics. So we thought if we could find a new fragment, that would be a good starting point for testing the feasibility of a generative AI approach. And we really wanted to do this comprehensively. So we assembled this database of 45 million chemical fragments that included 26 million fragments in the GDB11 database and 18 million fragments in the Antimicrobial database. And then we showed all 45 million of these fragments to our gonorrhea oracle, and it gave the thumbs up or thumbs down. And you can see the ones with high prediction scores made up the vast minority of compounds. Okay, so of the 45 million fragments, about 3 million were predicted to have some antibiotic activity against gonorrhea. We further filtered those to remove ones that were predicted to be cytotoxic to human cells. So we virtually counter-screened them with 3 human cytotoxicity models that we'd previously trained. We also removed any that had structural alerts, and we filtered out any structures that were similar to known antibiotics. And this left us with 1 million potentially interesting fragments, and that's obviously still too much. So we had to choose a method to try to whittle those numbers down further, since this was our first de novo design effort, and custom synthesis is both expensive and takes a lot of time. So we wanted to give ourselves the highest chance of success and iterate quickly. So we decided to first look to see, of those 1 million fragments, which are available in commercially available compounds, and can we order those compounds to actually experimentally validate their antibacterial activity in vitro first. Unfortunately, there were only 66 compounds that represented 27 of those 1 million fragments in commercially available space. When we tested those 66 compounds, 7 were antibacterial, and 2 that you see here, BRD1 and BRD2, shared the same fragment. And you can see here BRD1 and BRD2 structure, as well as their shared fragment called F1. And when we tested them, they weren't terribly potent at all, and they weren't very selective, but at least they were a starting point. So we continued with fragment F1, and we fed it into, we fed it into our 2 generative machine learning algorithms. The first is a genetic algorithm based on making chemically reasonable mutations, that's called CREM. And the second is a fragment-based variational autoencoder called the FVAE. So I'll first describe CREM. It's a genetic algorithm. You can think of it a bit as chemical evolution. It starts with your fragment of interest, and then it generates novel molecules by iteratively adding or replacing or deleting atoms and functional groups. And after each change, each round of change, we couple it to our graph neural network oracle, such that only the compounds that are scoring highly by our antibacterial prediction model and contains our original fragment would then be provided as an input to the next round of molecule generation. So we did these 5 rounds of evolution, or modifying the molecules, where each round kind of resulted, as you can see on this plot, of having progressively higher compound scores based on our ChemProc model, which is our oracle. Then there's a second generative model that was based on a fragment-based variational autoencoder model that was developed by our collaborator Wengang Jin. And this is based on a junctional tree variational autoencoder. Some of you might have heard of it. I won't go into the details, but basically it has an encoder model that takes your molecular graph of a chemical structure and transforms it into a latent vector. And then there's a decoder molecule that converts those latent vectors back into a molecular graph. And Wengang had trained this variational autoencoder on all 1.6 million bioactive drug-like molecules in the Chembl database, so that the model could learn what that drug-like space looks like. And then we gave it our fragment as an input. And the novel compounds were designed by the VAE by kind of sampling that latent space that was generated from the input fragment. Okay, so we took our fragment F1 that you saw, we fed it into both of the generative models. The CREM model made about 500,000 molecules, the FVAE made almost 7 million molecules, and then we filtered it for the predicted antibiotic activity using our antigonococcal model. Then we removed anything that was predicted to be cytotoxic to human cells. They had to be predicted to be synthesizable. Here we were relying on a couple different computational models for that. And then we wanted them to be structurally dissimilar, so we did another filter for diversity. And we were still left with hundreds of molecules. And RD had reached out to many chemical providers, synthesis providers, to see if these could be made. The number was still too big, so we whittled them down further, again, based on those that had the highest prediction scores using our antibacterial model and were most dissimilar to each other. That left us with 40 molecules. And of those, enamine was able to make 2 of our molecules of and it's only the ones that were designed by the FVAE. And interestingly, of those 2 molecules, which are shown here, which we called NG1 and NG2, they share that fragment that you saw before, fragment F1. NG1, right out the bat, had beautiful potency of 0.5 micrograms per mil against Neisseria gonorrhea, and NG2 didn't work at all. It wasn't effective at all against gonorrhea. So we had a 50% hit rate coming out of the gates. And we went on to test NG1 against for its spectrum of activity. We found that it was actually very narrow in its spectrum. It was only effective against pathogenic Neisseria, not even commensal Neisseria. And luckily, it retained activity against even some of the pan non-susceptible strains of gonorrhea that are available from the CDC isolate bank. And I'll come back to its mechanism of action later, but we can go back and talk about the second way in which we tried to design new antibiotics using Gen AI approaches, which was, what if you don't have a fragment at all? What if you let the models just go wild and design whatever they want without that constraint of an initial fragment? So we did this for Staph aureus this time. We tried our Staph aureus antibacterial prediction model that we used as our oracle to score the molecules that were generated by our 2 generative models of CREM and the JTVAE. For CREM, it needed some kind of starting point, so we gave it the most basic of ammonia, methane, or water to start building off of. For the JTVAE, we just ran it without a fragment constraint. And together, the models made over 29 million molecules, and the molecules that were made by the JTVAE model just looked more drug-like and more synthesizable. So we prioritized the JTVAE molecules. And it makes sense. I mean, it was trained on Chembl, so it learned what drug-like molecules really look like. And we did our same filtering that I described before for whether the Staph aureus antibacterial prediction model predicted that it was anti-staphylococcal. It had to look dissimilar from known antibiotics. It had to look like it was synthesizable by the RA score. And this time we were left with 4,800 molecules, which again was too much. This time we had the benefit of a chemist in our group named Andreas Lutens, who manually inspected these 4,000 molecules. And based on the building blocks that were available, he prioritized 90 molecules for synthesis, of which 22 could be successfully synthesized by enamine. So we tested those 22 molecules against Staph, and 6 were actually active, with a very high bar or low bar of an MIC of less than or equal to 64 micrograms. And their structures are shown here. And these compounds were selected because they had anti-staphylococcal activity. In most cases, they ended up having a broader spectrum of activity, mostly active against the gram-positives that we had tested, as well as Neisseria gonorrhea. For most of the gram-negatives that we tested, they didn't have activity on their own. It had to be adjuvanted with, let's say, polymyxin B treatment at the same time. And DN1 was one of these 6 kind of de novo designed molecules that looked like it was the most potent against Staph aureus, where it had MICs of 4 micrograms per mil and a decent selectivity index of 32. and it was able to maintain its efficacy against a panel of multi-drug resistant Staph aureus, including some visas and some linase lid resistant strains that we had tested. So this again gave us confidence that this approach of using graph neural networks as an oracle and pairing them with generative approaches could produce interesting antibacterial hits and be a starting point for drug discovery. So I'm sure some of you are wondering actually how these AI discovered compounds are actually killing bacteria. Of course, we were wondering that too. We really wanted to understand their mechanisms of action. We hypothesized that these mechanisms were different from existing antibiotics since they seem to be working against resistant strains. And we had many tools in our toolbox of trying to evolve resistant mutants using RNA-seq, looking for morphological changes and doing bacterial morphological profiling, as well as PISA. And I'll focus on NG1 and PISA really for the sake of time, because it proved to be a really useful technology for us. And okay, so with our collaborators at the Karolinska Institute, who are listed below, we used the proteome integral solubility alteration assay, otherwise known as PISA, to try to find possible protein targets for that fragment-based de novo design compound NG1 that I described first. And we saw some striking results. I guess stepping back, if you're not familiar with PISA, it's really a high throughput version of thermal proteome profiling. And it determines whether a small molecule is interacting with a protein by looking at the change in protein stability in the presence and absence of a small molecule. And it uses mass spec, and it assesses kind of protein stabilization changes on a proteome-wide scale. And the data for PISA can be visualized on a volcano plot like you would use for RNA-seq. And just like RNA-seq, we're looking for proteins with highly significant p-values and large fold changes. And when we tested that de novo design protein molecule NG1, the PISA assay really had this striking destabilization of the lipopolysaccharide transport system protein LPTA when we treated with NG1. And we performed some follow-up experiments that kind of supported LPTA as being the target. And I won't get into the experiments for the sake of time, but as many of you know, LPTA is an essential protein for the transport and assembly of LPS or LOS on the cell surface. And there have been antibacterial peptides like thanatin that have targeted LPTA, but to our knowledge, it hadn't been targeted by a small molecule before. And then finally, of course, we wanted to know whether it would work in animals. And after about a year of troubleshooting, we got the gonorrhea vaginal infection model working in our hands. And we found that when we treated with NG1 intraperitoneally, we didn't have a significant decrease in vaginal gonorrhea loads, but when delivered topically, we could see a significant decrease in gonorrhea titers within the vagina. And of course, there's still a lot of work to be done to try to improve systemic efficacy through analog design and SAR, and really developing ADMET ML models to better optimize these molecules up front for working in vivo. And I'll stop there and just give the takeaways, which I hope you now see that a deep learning model that's trained on phenotypic antibacterial data can enable efficient discovery and design of novel small molecules, and that the fragment-based generative design allows you to have a more systematic exploration of chemical space, where the VDs made nice drug-like molecules. And that synthesis is a challenge, but we hope that deep learning affords some increased efficiency gains in hit identification. There's a lot of chemical space left to explore. And I'll just leave up the last slide, which has all the people to thank, and Jim Collins's lab, as well as our funding sources and all of our collaborators. Thank you.
Very good. Thanks a lot, Melis, for this super interesting presentation. We now have about 5 minutes time for questions. So I've seen there are a couple that came in via the Q&A function. So please, others in the audience, make use of this Q&A function if you also would like to post some questions to Melis. So perhaps let's start with one that Pavel posted here in the chat. So in essence, what he's asking is that obviously you needed to down select and quite aggressively because there's only so few that you can actually validate and synthesize, right? Now, his question is, how could you, or do you, do you have an idea of your false negative rate? So in a way, could you somehow try to figure out what you're missing on, because the AI, has not scored a molecule that actually is active properly?
The false negative rate. Yeah, I mean, it is certainly possible that we have, as you saw, many steps in our filtration process. Synthesizability, I think, is the one where perhaps we have the greatest false attrition because those models are just really difficult, and we know there's a lot of improvement to be made there. So it is possible that we are removing compounds that are really antibacterial. We haven't looked too much at the false negative rate. We did one experiment with an internal library within the Broad Institute that has 800,000 compounds, and this is in the paper that's currently under review, I hope will come out soon, where we took those 800,000 compounds, we scored them with the gonorrhea model, and we also took a random selection of 30 molecules. And with our models, we were getting anywhere between 10 and 80% hit rates. With the random selection, we had 0% hit rate. So that is not exactly answering your question, because we didn't actually go and then test the entire 800,000 compound library for time and cost reasons. But I think that the model itself is more likely to have false positives than false negatives, unless the compound that you're trying to make that prediction on is very different from the compounds that are within your training set. And in that case, I think you're very likely to get a false negative, because the model has just not seen an example of an antibacterial compound that looks like the compound you're showing it. So I would say the false negative rate is probably highly dependent on the chemical structures of the compounds that you're showing it and using the model to score.
Okay, very good. Thanks, Melis. So perhaps one more question, and just as a quick piece of information for the audience. So Victor and I agreed that we will collect the questions that we cannot answer in this session and forward them to the presenters. So as long as it's not a couple of hundred, we're hopeful that that our presenters are kind enough to maybe comment on a few extra ones offline. So just for your information. Perhaps for the last 2 minutes, maybe I can try to condense a couple of questions regarding the positive selection into one final question for you, Melis. So I like your analogy with the function, the AI actually being a function that translates a molecule space, a chemical space, into a number between 0 and 1. And I was actually intrigued to see that even in the data you already presented, it looked more like a vector function, if you wish, because you're not only looking for efficacy, but you're also looking for synthesis, the ability to synthesize the drug. And then along these lines, I guess, am I right when I say that where we want to be is probably a situation where we can further down select the compounds we eventually synthesize, and I'm particularly thinking about the potential for drug-drug interactions. Somebody else brought up the potential for resistance development. So am I right when I say that this is sort of the frontier right now, and this is kind of, you could factor all this stuff in and then have a more and more intelligent approach, basically, and that's what we're trying to achieve next? Is this statement correct?
Perfect. Yeah, completely agree. I think this was the first step where we did this iterative filtering approach, where the first step was just purely antibacterial prediction. But I completely agree with you. The ideal would be that you had some kind of multimodal model that takes into account everything that you want in the final drug and does that optimization up front, rather than potentially losing compounds through that funnel or filtration process. So I know there's a lot of active work on being able to do that upfront optimization, and hopefully we'll see that coming out in the next few years. But that's definitely the direction that this is moving in. I totally agree.
Very good. Thanks a lot, Melis. Super interesting work, and we're, I guess, all very keen to see what comes next. So again, in the interest of time, apologies. I've seen there are a couple of extra questions, but as I said, we will capture them and then forward to Melis. Thanks a lot once again for your super nice presentation. And I think now it's time to move on to our final speaker, Nicole. So welcome, Nicole, to this webinar. Nicole Scangarella-Oman is the scientific director in infectious disease research at GlaxoSmithKline in Collegeville in the US. She joined the clinical microbiology group at GSK, and for over 2 decades now has focused on clinical microbiology and the development of GSK antibacterials. She's currently leading microbiology for a late-stage antibacterial. Nicole is also a member of the CLSI-AST subcommittee, ESCMID and ASM, and is the author of over 50 peer-reviewed scientific publications and approximately 140 scientific abstracts. So welcome, Nicole, once again, and the floor is yours.
Thank you. Thank you for the introduction. Good afternoon, and thank you all for attending this webinar, and many thanks to the organizers for allowing me the opportunity to present the findings from our manuscript, which was published in Antimicrobial Agents and Chemotherapy in 2025, and outlines microbiology data for gepotidacin from 2 global pivotal phase 3 trials for the treatment of uncomplicated urinary tract infections. Next slide, please. So first, some disclosures and disclaimers. I am Nicole Scangarella-Oman, as mentioned, as Scientific Director at GSK, and I am an employee of GSK. Some additional disclosures and disclaimers are shown on this slide. But importantly, the data I will be sharing was in part funded by federal funds from the United States Office of the Assistant Secretary for Preparedness and Response, Biomedical Advanced Research and Development Authority, otherwise known as BARDA. So next slide, please. To start, I'd like to provide some brief background regarding gepotidacin and also uncomplicated urinary tract infections. As many are aware, UTIs are among the most common bacterial infections encountered worldwide, and approximately 50 to 60% of adult females will experience at least one urinary tract infection in their lifetime. The diversity of virulence factors and evolving resistance mechanisms in uropathogens help to contribute to rising antimicrobial resistance, which limits available options for uncomplicated UTI treatment. To help address this, gepotidacin was designed as a potential alternative oral treatment option for women suffering from uncomplicated UTIs. So what is gepotidacin? So gepotidacin is a novel bactericidal, first-in-class triaze-acinaptyline antibacterial that inhibits bacterial DNA replication by a distinct binding site, a unique mechanism of action, and for most pathogens, provides well-balanced inhibition of 2 different type 2 topoisomerase enzymes. This provides activity against most uropathogens, including isolates resistant to current antibacterials. Gepotidacin was evaluated for the treatment of uncomplicated UTIs in 2 recently completed global phase 3 trials, EAGLE-2 and EAGLE-3, which were conducted between 2019 to 2023. Based on the results of EAGLE-2 and EAGLE-3, gepotidacin was approved in March 2025 by the United States Food and Drug Administration for the treatment of uncomplicated UTI. Next slide, please. Now I will briefly describe the phase 3 uncomplicated UTI clinical trials. EAGLE-2 and EAGLE-3 were global phase 3 double-blind, non-inferiority, randomized controlled trials. Both studies were conducted in accordance with current FDA and EMA guidance for uncomplicated UTI and were designed to demonstrate the non-inferiority of oral gepotidacin, 1,500 milligrams administered twice a day for 5 days, to oral nitrofurantoin, 100 milligrams administered twice a day and also for 5 days. To be eligible for each study, participants needed to be female, over 12 years of age, have at least 2 symptoms of uncomplicated UTI, and evidence of urinary nitrate and/or pyuria. Each participant attended 4 scheduled visits, baseline, an on-therapy visit between days 2 to 4, a test of cure visit between days 10 to 13, and a follow-up visit at around day 28. The primary analysis population included all randomized participants who received at least 1 dose of study treatment and had 1 or 2 baseline qualifying uropathogens that were also all susceptible to nitrofurantoin per CLSI guidelines. Qualifying was defined as uropathogen growth of greater than or equal to 10 to the 5 CFUs per ml. This population was otherwise known as the microbiological intent to treat nitrofurantoin susceptible population, or for short, micro-ITT NTFS. The primary efficacy endpoint was defined as therapeutic response at the test to cure visit. Therapeutic response is a composite endpoint of both clinical and microbiological response, the definitions of which you can see on this slide. Primary results from both studies have been previously reported, and in each phase 3 trial, gepotidacin demonstrated non-inferiority to nitrofurantoin, and in EAGLE-3 also demonstrated superiority. Next slide, please. So in the next few slides, I will show pooled microbiology results and efficacy observations from EAGLE-2 and EAGLE-3 with a focus on overall incidence of recovered uropathogen and drug-resistant phenotypes, gepotidacins in vitro activity, and bipathogen efficacy responses against uropathogens and drug-resistant phenotypes. Now, in contrast to the primary analysis I just mentioned on the prior slide, for the micro-ITT NTFS population, which only included micro-ITT participants with nitrofurantoin susceptible baseline qualifying uropathogens. The data in the next few slides are based on the micro-ITT population, which includes all patients that had a baseline qualifying uropathogen regardless of susceptibility to nitrofurantoin. The rationale for utilizing this population, as it included a higher number of participants and uropathogen data, thus providing a broader and more clinically relevant data set for assessments. To briefly describe the microbiological assessments from these studies, participants provided a midstream clean catch urine sample at each visit, which was then transported to a central laboratory for quantitative urine culture and identification using standard methods, and then subsequent antimicrobial susceptibility testing according to CLSI guidelines. It should be noted that the pooled EAGLE-2 and EAGLE-3 uropathogen data is distinct from other data sets for community-acquired uncomplicated UTIs, as they are based on urine specimens collected from all participants without selection bias. Other databases typically contain cultures performed for selected patients based on past medical history or recurrence of uncomplicated UTIs, whereas our approach has led to a comprehensive and unbiased set of contemporary uropathogen prevalence data in females with uncomplicated UTI. Next slide, please. On this slide is a table describing the incidence of baseline qualifying uropathogens in the micro-ITT population from both trials. Among micro-ITT participants, 1,486 baseline qualifying uropathogens were recovered. The most common identified species was E. coli at 78%, followed by Klebsiella pneumoniae at 8%, Proteus mirabilis at 5%, and Staph cephalopoticus at 2%. All other baseline qualifying uropathogens identified had an incidence of less than 1%. The specimens recovered and overall prevalence are consistent with known uncomplicated UTI uropathogen epidemiology. Next slide, please. And shown here is a table describing the incidence of the selected drug-resistant phenotypes for E. coli and Klebsiella pneumoniae that were seen in both trials. Among 1,159 E. coli isolates in the pooled micro-ITT population, resistance to fluoroquinolones or trimethoprim sulfamethoxazole was 28% for each. 28% of isolates were multi-drug resistant, and 15% were extended spectrum beta-lactamase positive. With the exception of trimethoprim sulfamethoxazole, the percentage of E. coli isolates resistant to first-line recommended treatments was low and ranged from 1 to 3% for fosfomycin, nitrofurantoin, and methicillinum. For the 114 baseline qualifying Klebsiella pneumoniae isolates, The most common drug-resistant phenotypes were nitrofurantoin resistance at 25%, 24% were multi-drug resistant, and 23% trimethoprim sulfamethoxazole resistant, followed by fluoroquinolone resistance and ESBL positive isolates, each were at 16%. No baseline qualifying E. coli or Klebsiella pneumoniae isolates were resistant to meropenem. And available data for additional drug-resistant phenotypes for other uropathogens, such as Proteus mirabilis, can be found in the manuscript. Next slide, please. So next, I will describe gepotidacin activity against the uropathogens and drug-resistant phenotypes that we saw in the pooled micro-ITT population. Shown on this slide are the range of gepotidacin MICs, MIC50s, MIC90s, and also percent susceptibility for selected species and drug resistance phenotypes of E. coli and Klebsiella pneumoniae. Percent susceptibility for gepotidacin is based on the recently FDA-approved breakpoints and interpretive criteria for Enterococcus, Staph saprophyticus, and Enterococcus faecalis. Based on these FDA breakpoints and interpretive criteria for gepotidacin, the vast majority, 93 to 100% of baseline qualifying uropathogens were susceptible to gepotidacin, and no Enterobacteriaceae isolates were resistant to gepotidacin. Additionally, as shown in the table, for all uropathogen drug-resistant phenotypes, gepotidacin MIC90 values and percent susceptibilities were generally similar when compared with the MIC90 and percent susceptibilities of the overall species, demonstrating the in vitro activity of gepotidacin. Specifically for E. coli, greater than 99.9% of E. coli isolates were susceptible, and the percentage of E. coli isolates susceptible to gepotidacin for the drug-resistant phenotype shown ranged from 96.9% to 100%. For Klebsiella pneumoniae, 96.5% of all isolates were susceptible, and percent susceptibilities ranged from 85.7% to 100% for the drug-resistant phenotype shown. Available in vitro activity data against drug-resistant phenotypes for other uropathogens, again, can also be found in the manuscript. Next slide, please. So these next three slides will present forest plots of therapeutic, clinical, and microbiological success at test to cure by baseline qualifying uropathogen and selected drug-resistant phenotypes in the pooled micro-ITT population. On this slide is data for therapeutic success. As can be seen from the figure, within the gepotidacin treatment group, therapeutic success rates were generally similar across phenotypic subgroups of E. coli. And even though sample sizes were small for Klebsiella pneumoniae and Proteus mirabilis, drug-resistant phenotypes, gepotidacin success rates for each phenotype were consistent with the corresponding therapeutic success rates for each overall species. Generally, and as expected, therapeutic success rates were numerically higher in the gepotidacin group compared with nitrofurantoin group, since this analysis population did include isolates that were not susceptible to nitrofurantoin. Next slide, please. On this slide, shown is a forest plot with data for clinical success by baseline qualifying uropathogen and selected drug-resistant phenotypes at test to cure for the micro-ITT population. While many clinical success rates were numerically higher than the therapeutic success rate shown on the prior slide, we saw similar trends seen as gepotidacin clinical success rates were also primarily consistent across phenotypic subgroups of the species shown. And again, for the reasons I just stated previously, generally and as expected, clinical success was numerically higher in the gepotidacin group compared with the nitrofurantoin group. Next slide, please. And last but not least, this slide shows a forest plot illustrating microbiological success at test to cure by baseline qualifying uropathogen and selected drug-resistant phenotypes for the micro-ITT population. Similar to what I just stated for clinical success, while many microbiological success rates were generally numerically higher than both the therapeutic and clinical successes, similar trends overall were seen, As within the gepotidacin treatment group, the microbiological success was generally consistent among phenotypic subgroups of each species. Although again, sample sizes were small for some E. coli and all Klebsiella pneumoniae and Proteus mirabilis phenotypic subcategories. And again, generally and as expected, microbiological success was numerically higher in the gepotidacin group compared with the nitrofurantoin group, as this analysis population did include isolates not susceptible to gepotidacin. Next slide, please. So in summary, the microbiological data presented from EAGLE-2 and EAGLE-3 demonstrated a number of aspects, including that consistent with known UUTI pathogen epidemiology, E. coli was the most prevalent pathogen recovered, and resistance to current treatment options for uncomplicated UTIs was observed in this outpatient population, with drug-resistant rates greater than 10% for some drugs. Per FDA, gepotidacin breakpoints and interpretive criteria, the vast majority of baseline isolates were susceptible, and no Enterobacterales isolates were resistant to gepotidacin. And for all uropathogen drug-resistant phenotypes, gepotidacin MIC values and percent susceptibilities were generally similar when compared with the MIC 90s and percent susceptibilities of the overall species, again, demonstrating the in vitro activity of gepotidacin. And finally, with regards to efficacy, therapeutic, clinical, and microbiological success for gepotidacin attested cure in the micro-ITT population were generally consistent across phenotypic subcategories of E. coli, Klebsiella pneumoniae, and Proteus mirabilis. So in conclusion, the pooled microbiology data from the EAGLE-2 and EAGLE-3 pivotal global phase 3 trials provided a contemporary, comprehensive set of prevalence data for community-acquired uncomplicated UTIs in females from an unbiased data set, and also provided evidence supporting the in vitro activity and in vivo efficacy of gepotidacin against uropathogens and drug-resistant phenotypes relevant for the treatment of uncomplicated UTIs. Next slide, please. I would like to specifically thank the patients, their families, their caregivers, the clinical trial investigators, and all their team members at each site who participated in the EAGLE-2 and EAGLE-3 trials. I would also like to thank my co-authors on this manuscript. And as previously mentioned, the studies described in this presentation were funded in part by BARDA. And with that, that's my last slide. So many thanks for attending and for listening about the results from our pivotal phase 3 trials for gepotidacin. And I will take any questions that you may have.
Thanks a lot, Nicole, for this super nice presentation and very interesting and strong data. So I just had a look at the Q&A, and once again, audience, this is your last chance to make use of the Q&A function. This time specifically for Nicole, and Pavel is first here on my list, and he writes very nice data. I fully agree. And his question is, would you expect efficacy to remain as strong in more complicated UTI settings, recurrent infections, or patients with prior antibiotic exposure, where bacterial populations may be more heterogeneous than in uncomplicated UTI?
So the current, thank you for that, was a great question. The current indication for gepotidacin is specific to uncomplicated UTI. We did not study patients with complicated UTI, which obviously involves situations outside of the bladder. We do have some data that was presented previously in other manuscripts regarding various subgroups, like those with recurrence and various other factors. But for complicated UTI, gepotidacin is highly concentrated in the urine, so we do not anticipate that it would be efficacious in those body sites outside of the urine for this indication.
Thanks, Nicole, for this very clear answer. And there's another question that just came in by Karine, and her ask is if you would maybe just summarize the main difference between gepotidacin and other antibiotics typically used in this indication. What makes it new, novel, special?
Yeah, thank you again for another great question. As we mentioned on the first slide, gepotidacin is unique. First of all, it's first in class, so it's a new class of antimicrobials, the triaza-acinaphthalines, and it inhibits the DNA bacterial replication at a unique, distinct binding site. So the binding site for gepotidacin is different from for quinolones and other antibacterials. It also has a novel mechanism of action of how it inhibits the bacterial DNA replication. And it also has the ability to have well-balanced targeting of 2 different enzymes. So that then increases the potential barrier for resistance development.
Very good. Thanks for this very clear summary. Now, my question to the audience, do we have any other questions? Anything else coming up? That does not seem to be the case. Okay, very good. So thank you once again, Nicole, for this great presentation and this great talk. So that means we're at the end of the presentations for now. And this also means that I will pass it back to you, Victor, for your closing remarks. From my side, I would already like to thank both the speakers and then everyone in the audience for this really nice presentation, for this really nice session. Sorry, I think we had heard 3 really nice presentations with lots of interesting data spanning a wide scope of topics. And yeah, Victor, back to you. Let's see if we want to continue with a similar format or not. And thanks a lot to everyone from my end.
Sure. Thanks so much, Florian, for moderating today. So just to repeat what Florian said before, we'll be asking the speakers to provide written responses to the questions that weren't answered in the webinar, and we'll make this available on the Revive website, revive.gardp.org/webinars, where you can see recordings of our previous webinars as well. So thank you also, David, Melis, and Nicole, for your interesting presentations. I'd like to also now announce our next Revive webinar, which you can already register for. This will be on the 28th of April, and this will be on the successes and future prospects of natural products inspired antibiotics. So please keep an eye on the Revive website to hear more news from us. And also you can subscribe to our newsletter there and follow GARDP on social media to hear more of our updates and announcements. So that will be all from us today. Thank you for joining and contributing to this discussion. I hope you found the webinar interesting and useful and that you will join us again in the future. Please do spread the word amongst your colleagues as well to join next time. Thanks again and goodbye.